open some of my data here. So we're going to talk about microRNAs. I'm not sure how much you know about microRNAs. I will try to explain just a bit. Okay, one of the most striking features in the evolution of genomes is the observation that the more complex an organism is, the larger amount of non-coding protein sequences they have in the genome. In fact, in humans, only 2% of our genome code for proteins. And that means that 98% are non-protein coded sequences. Some recent data from the Encyclopedia of DNA Elements say that 85% of our genome has a function, at least biochemically speaking. And from that, around 76% is transcribed. Okay, if we have just only 2% of our genome that transcribed for proteins and 76% from all is transcribed, so what is the rest? That's the question. So we now know that our genome uses a lot of the information to codify for a large number of non-coding RNAs. Among non-coding RNAs, we have small RNAs, we have RNAs, long intergenic non-coding RNAs, pig interacting RNAs. So we can consider our eukaryotic genome as an RNA machine. So talking about the family of small RNAs, we have the most common are three of them: are small interferon RNA, we have pig interacting RNA, and microRNAs. All are smaller than 30 nucleotides long. So we are going to focus only on microRNAs. MicroRNAs were first discovered in 1993 while studying the animal model C. elegans. They observed, for instance, when the larval stage L1 to L2, just to make this possible, there need to be a reduction of two proteins, which was lin14 and lin20A. But they didn't know how this was possible. So while doing a screen, they find out that there was a small RNA molecule that increased like crazy. So they called this one lin4. Then in this study, it was a back-to-back paper in a Cell. They found that these two proteins, lin14 and lin20A, you can see here, both have a complementary binding sequence for this small RNA, which was called lin4. So they demonstrated that year for the first time that in our genome have a small RNAs that could modulate the expression of different genes. It was not until 2001 that people demonstrate that this phenomenon was not only for C. elegans, but was for different species, including humans and plants. They call microRNAs to this family of RNAs. So microRNAs are synthesized from our genome by RNase polymerase II. They synthesize the prime microRNA, which is around 1,000 kilobase long. Inside the nucleus, they are cleavaged by Drosha and Pasha, generating the pre-microRNA, which is around 70 and 120 nucleotides long. Then the pre-microRNA goes out from the nucleus to a cytoplasm by the protein exporting 5. In the cytoplasm, these microRNAs bind to another enzyme, a ribonuclease type 3, which is called dicer. Dicer cleavage the hairpin structure and generate a double-strand small RNA from around 21 nucleotides. From one of the strands is degraded, and the other one is the mature microRNA isoform that bind to the argonauts proteins, and from here guide the RISC complex to the 3' UTR of their targets to produce an inhibition of the translation or degradation of the mRNA. So the final function is to reduce the protein output. Okay, one of the most interesting functions of microRNA are two characteristics. The first one is that, for instance, this is the 3' UTR of any gene, insulin receptor substrate 2, which is involved in insulin metabolism. Okay, so several microRNAs, different microRNAs, could regulate this protein post-transcriptional, this mRNA. So this is very interesting. The other thing, which is also much more interesting, is that one microRNA could regulate at the same time different proteins. So that means that if you are able to modify the expression of this microRNA, either by pharmacologically or by any dietary intervention, you could modulate at the same time different genes. And it is very interesting if all those genes, or some of them, are within one pathway. So that means that you could modulate the entire pathway. So in the last decade, there have been several advances in the field of microRNAs. For instance, these are just only some examples of microRNAs that are involved in cardiovascular medicines. For instance, this microRNA 143, 145 regulate the phenotype is switch from proliferative to contractive vascularized smooth muscle cell and regulate vascular tone. It's very important in hypertension. There are other microRNAs that are involved in glucose metabolism. For instance, microRNA 375, LED7, 103, 107, 143. They regulate different proteins involved in glucose metabolism. And we're talking about lipid metabolism, for instance, microRNA 33 regulate both oxidation of fatty acid by regulating different proteins involved in fatty acid beta oxidation, and also regulate cholesterol efflux and HDL by regulating ABCA1. This is localized within one intron of one of the most important transcription factor about lipid metabolism, which are the SRBP family. MicroRNA 122, which was one of the first microRNA discovered and studied, regulates several aspects of lipid metabolism and cholesterol metabolism. Something interesting about microRNAs is also that they can be secreted from one cell and then have their action in another different cell. They are secreted by multivesicular bodies, by exosomes, or even by lipoproteins. They are uptaken by a different cell line and they exert their function. It's kind of like an exocrine function. And as all the microbiome that are everywhere, also microRNAs have been found in almost every single biological fluid. You have everywhere are microRNAs secreted. We still don't know why they are there and what's the real function of them. But people are using circulating microRNAs to use like a biomarkers of disease. For instance, there are several microRNAs that are related to different types of cardiovascular disease. So here came the most important. So even in the last 10 years, the pharmacological research of microRNAs have increased dramatically. For instance, this is an antisense oligonucleotide. What you do is, so this is the mature isoform of the microRNA, for instance. Then you generate an antisense sequence from the same RNAs, and then you do different conjugations. You include a fluor here, a cholesterol to increase the capture by the liver, or you increase different chemical modification to increase the half-life of this antisense molecule. In fact, last year came out the first clinical study of the first microRNA, which is against microRNA 122, for the treatment of hepatitis C virus infection. And the results are very, very good, very promising. And there are several other microRNAs that are already, some in very advanced preclinical studies and some others in clinical phase. For instance, this miR122 is the first one, but this second one, which is already in clinical studies, is miR34. Apparently, you lose this microRNA in certain type of cancer. So if you put this microRNA inside in the humans, then you are supposed to treat a certain type of cancers. So the question is, so we can manipulate microRNA function or activity pharmacologically. Can we do this also with dietary components? And the answer is yes. For instance, I will show you just only some examples of how this can be possible. There are increasing number of studies that say this. For instance, we were trying to see if the consumption of DHA, this fatty acid from fish oil, could regulate the expression of microRNAs. Okay, so these are intestinal cells exposed to DHA lipid micelles, and we evaluate the whole genome of microRNAs, and we found that there are certain microRNAs that were modulated by this fatty acid, but not the other ones. So that looked like maybe kind of specific. So we found these two microRNAs, microRNA 30C, and when we see all the targets of this microRNA, we could see very nice targets related to lipid metabolism, including microsomal triglyceride transfer protein, LDL receptor, ABCA1, VLDL receptor, and also this other microRNA, which is the microRNA 192, also regulate different key genes involved in lipid metabolism. For instance, ABCG4, caveolin-1, caveolin-2, also the transcription factor of lipid metabolism, SRBF1, ABCG5, also LDL receptor. So this may explain in part why when you drink or when you eat fish oil, you have reduced level of triglycerides. This is one of the mechanisms we explain through microRNAs. In this other study, we tried to do something different. There was some colleagues that they observed that when they give different types of fatty acid from diets, there were 5 types of fatty acid. Sunflower oil, olive oil is the second one. The third one is fish oil, linseed oil, and palmitic oil. So they give just only during the first 12 days of pregnancy of the rats. And they observed that just only given that 12th first day and then thereafter they give a normal diet, and they evaluate the offspring at 4 months and 8 months, and they observe that those ones that have fish oil during the first 12 days of the pregnancy, they have high insulin sensitivity index. So they didn't know why this was possible, what was the explanation. So we started to see whether microRNAs could play a role in this. So we evaluate microRNAs in all the process, and we find out that those rats that consume fish oil here, they have a lower expression of 2 microRNAs, which was the microRNA 10B and 192, and some of the very important genes related to insulin signaling are target of this. You can see here that are increased by this treatment. So maybe the explanation is that this fish oil do something epigenetically. So when the mothers have fish oil, the offspring, when they are adults, they have a lower expression of this microRNA, and in that form explain why they have more insulin sensitivity index. In this other study, it was a clinical trial. We give to one group a placebo, another one grapeseed extract, and to other group grapeseed extract plus resveratrol. And we find out that during 3 times, we took some samples at baseline at 6 months and 12 months, and we isolate polymorphonuclear cells from circulation. And we show, for instance, that those ones that consume grapeseed extract plus resveratrol, they have higher level of microRNA 21 and 181. And if we evaluate target of these 2 microRNAs, they have very nice target related to inflammation. So explaining why people that consume this extract with resveratrol have a lower inflammation index in the circulation. So like that, we are conducting several studies to see if we could find any microRNAs that could be regulated specifically by a dietary polyphenol. For instance, in this study, we are just evaluating where the hydroxytyrosol, which is the main polyphenol of olive oil, could regulate all these microRNAs. And if one of these is exclusively from this one, also with urolitins, which is the major metabolite from those ones that consume pomegranate juice, you have nice changes of different microRNAs. And also, these are rats consuming, this is in humans. This is in mouse, and this is in rats consuming an extract of Rosmarinus officinalis. In the liver, you see also very nice changes. So then, it is possible to modulate the expression of microRNAs through the diet. The question is, which one of these is specifically, so you can you can use this therapeutically. So we are trying to study some of them to see which of them are really specific for this microRNA. So in summary, the function of microRNA, when one microRNA binds to the 3' UTR of a messenger, they reduce the translation or repression of the mRNA. So they reduce the synthesis of proteins. Okay, there is one kind of therapy is when you include an extracellular, this microRNA, but from an extracellular source. So this is the microRNA replacement therapy, just to reduce the expression of this protein. Maybe in some diseases this protein may be upregulated, so you can use this microRNA mimic, or you can use also a small molecule activator of the expression of the microRNAs. So this is the 2 possibilities. And the other possibilities is when one microRNA is increased in a disease, so what you want is to reduce their expression, so then you can use antisense oligonucleotides, and you can also say microRNA masking. You can use microRNA sponges, so this is another sequence where you insert to the cell, and then this sequence captures all the microRNAs, so they don't bind to the 3' UTR of the gene, so their expression increase. And also another type of RNAs, long noncoding RNAs, which are circular RNAs that have several binding sites. So in conclusion, it's feasible to modulate the expression of microRNAs, and I think this may explain why when you have a healthy diet, maybe you are regulating the expression of several genes by their own, by also through these microRNAs. And I want to finish just giving the acknowledgement to all these people. Thank you.